My name is Cian Schmitt-Ulms, and I'm a member of Jonathan Gootenberg and Omar Abudayyeh's lab, mostly working on CRISPR systems and how we can employ them for different therapeutic applications. So the talk I'm going to give today is called Leveraging DNA Targeting CRISPR Systems for Genetic Editing. And more specifically, I'm going to talk about a project that we published last year that is employing a couple of different systems from different parts of biology to create a very desirable type of genetic editing. So the tool that we've been developing is called PASTE. And the purpose of this tool is to generate site-specific large DNA integration events. So if we look at the recent history of genetic editing and gene therapy, we've seen a gigantic range of really editing modalities that have been made possible by these RNA-guided CRISPR-Cas systems that were developed in the early 2000s and 2010s. And really, starting from at the most basic systems that cut DNA, people have been developing additions and modifications to the basic CRISPR-Cas system to create a variety of different applications and tools, including things like CRISPR systems that up or down regulate a specific gene, or bring parts of the chromosome closer together, or for the purpose of imaging, or create histone modifications, or otherwise remodel the genome. And a lot of these systems really originate with bacterial defense systems. So these are systems that bacteria employ to defend themselves against invasion, usually from viruses. So in the case of CRISPR-Cas9 or CRISPR guided by RNA, you have a virus that is initially invading a bacteria, and a bacteria that is able to recognize it using this spacer sequence that's complementary to the virus and cut it. And we've been leveraging this in, I think, a pretty interesting way that I'll talk about. So before I get into the actual tool, I just want to briefly mention that when it comes to the sort of realm of therapeutic edits or therapeutic applications, there's already quite a lot that's possible. Basic CRISPR or CRISPR modified or conjugated with base editing modalities can create these single base changes, whereas tools that are a little bit more complicated, like prime editors, can address a variety of small modifications in DNA, essentially inserting or removing or slightly adjusting individual bases or small groups of bases. So where we see a gap is actually in the realm of very large modifications. So there's a lot of cases where it's not enough to just change one base at a time or insert a couple of bases into a gene of interest. And in reality, the solution to the problem requires a very large insertion or the replacement of an entire region. So in the past, what people have been trying to employ are generally tools that will cut the genome and then try to insert based on a templating known as homology-directed repair, a large sequence. The problem with a lot of these approaches is that they have a very high potential for causing damage. When people talk about DNA damage, normally in the form of indels, which are insertions or deletions, but really any event where you cut and insert and you get an outcome outside of what your desired edit is could be considered an indel. And so what we need to develop is a tool that allows us to make large insertions while reducing the amount of damage and reducing the potential off-target effects that can come from these sort of blunt cuts created by basic Cas9 editing. So to address these issues, we've developed a tool that combines some earlier applications such as prime editing with some knowledge that was already existent about the function of integrases. And what integrases do is effectively recognize a piece of DNA and integrate another piece of DNA into it. And so what we've done is take a prime editor and we direct a prime edit insertion towards a site of interest. the purpose of this edit is to install a landing site that an integrase can then recognize. So we have this two-step process where we use CRISPR to effectively create a landing site, and then we use an integrase, another bacterial system, to install our edit into it. And by combining these two systems, we can sort of get the benefits of both, where we get a very specific targeted insertion, and then immediately afterwards, a very large integration into that position. So when I say large, we've tested at this stage using this PACE system, insertions of up to 36 kilobases, and really very consistently insertions of greater than 2 or 4 or 5 kilobases. And so we're dramatically increasing the sort of scale of edits that are possible. And with this capability, we can potentially address a lot of the problems that are previously not resolvable by tools like base editing or prime editing, by simply replacing gigantic regions of the genome at will. So in the course of generating this PACE system, we underwent several rounds of engineering. And really, when you have a system that is a little bit more complicated to the degree that PASTE is, you have a lot of different angles of approach for engineering. So since, in effect, PASTE is a two-step process that requires both a prime editing event and an integration event, we have a couple of different angles, including trying to enhance the efficiency of the integrase, trying to enhance the efficiency of the prime edit, and then the efficiency of the prime edit in turn being dependent on the efficiency of both targeting and reverse transcription. And we've gone about this sort of engineering approach through a couple of different ways, including mining of different orthologs of integrases, which I'll talk about in a moment. At its most basic, before I get into the integrase searching, we are able to engineer or improve the efficiency of the PACE system simply by changing the way the individual subunits are connected to each other. In this case, I'm showing some data where we generate a whole set of fused constructs that have different linker regions in between the prime editor and the integrase, and explore how the structure of these different linkers affects the overall efficiency of the system at a variety of different positions. What I'm also showing here is that we spent some time trying to engineer the PEG RNA, or the scaffold effectively for PASTE that is responsible for the initial prime edit. And what we find is that by both of these approaches, we can generate a pretty large enhancement in the overall efficiency. So this is one of the, I would say, simpler approaches for improving efficiency. Beyond that, we wanted to spend some time trying to understand what factors affected the initial prime editing efficiency. Because PASTE is dependent on an initial prime edit, we need to really try to enhance each of the individual steps to get the greatest maximum overall rate. What we did in order to look at the prime editing step is generate a whole set or a library of different prime editing guides that combine different sort of design rules about the sizes of the individual components. So looking at both the size of the attachment site that we insert, the size of the RT template, or of the PBS strand, and by co-varying all of these in a lenti screen, we were able to identify a set of design rules for a set of 8 endogenous genes that dramatically boost the efficiency. One of the problems that we identified initially is that the insertion efficiency seems to be relatively locus dependent. So due to the fact that different genes or different positions in the genome have different structures, what works in one position, such as ACDB, may not be the ultimate pegRNA construct design for another gene, such as LMNB1. What I would say is that this type of pooled screening sort of illuminates how there are a whole set of different levers that you can pull on if you're trying to create like an ultimate optimized construct for any particular target. So going briefly into the other side of PASTE, which is following the prime edit, we have to integrate our cargo. The way that we are integrating this cargo is by using an extremely efficient enzyme called the BXB1 integrase, which is a large serine recombinase. And what this enzyme does is recognize a @B site, which normally would be an attachment site found in a bacterial genome, and an @P site, which you can think of as its plasmid attachment site. And by recognizing these two sites, it will bind, bring them close together, and then undergo sort of a translocation or a rotation event where it joins together the @P and @B site and creates a insertion event. We were able to probe some of the characteristics of how BXB1 recognizes its site and what factors affect its efficiency by generating a whole set of mutants of both the @P and @Bs. Similar to our guide library screening, we generated a library of @Ps, which are effectively different variants of the attachment site of our cargo or of the inserted DNA, and probe them for efficiency. By doing this, we were able to identify that the efficiency of @P insertion into an @B is particularly dependent on the identity of the bases in the middle of these sites. And what we were actually able to find out is that BXB1 really does not like to integrate a mismatched @P cargo into an @B site that doesn't have a complementary @B. And by using this characteristic of BXB1, we're able to generate a whole set of orthogonal pairs of @Bs and @Ps that we can use to orthogonally and in parallel integrate into a variety of different sites. So what I'm actually showing here is that by transfecting at the same time a set of guides and cargoes that target different genes, including ACDB, LMNB1, and NULC1, we can concurrently edit at 3 different sites at the same time with relatively high efficiency. This might be interesting in cases where you're trying to use a system like PASTE to label a set of transcripts or a set of genes at the same time, or just in general exposes how specific the BXB1 integrase used is really for its on-target. We were interested in seeing whether we could further improve the efficiency of PASTE by swapping out BXB1. So while BXB1 is an incredibly efficient integrase, we thought there might be other members of its family or other families of integrases or recombinases with higher efficiency. So we did a computational search for different novel integrases based on similarity to the BXB1 sequence, and discovered a whole set of thousands of different integrases and tried to estimate their efficiency. We then grouped the integrases that we discovered into a set of different families and took representative integrases from each of these families to see if we could find something that had higher efficiency than BXB1. One of these integrases that we ultimately selected is from B. cereus. And we found that by combining the B. cereus integrase in the place of BXB1 integrase, for our paste system, we could actually enhance the overall paste efficiency at several genes, including actin beta, where in this panel on the right, we got almost twice the editing with the swapped out integrase. One of the things I want to focus on is that the paste system, relative to some of the other approaches people have been using, namely Cas9 cleavage and HDR insertion, the paste system has a higher overall rate and lower amount of DNA damage. So when we compare paste off-target damage and paste indel rates at a variety of different loci, we find a favorable outcome. This is incredibly important because anytime that you introduce a DNA targeting system, especially in the realm of a therapeutic application, you have to be concerned about the potential of an off-target integration or off-target cleavage. By not relying on double-strand breaks, we avoid a lot of the concerns about indels, and by using an incredibly specific BXB1 integrase for the insertion, we can sort of guarantee that we have almost no off-target insertions of our cargo, which could cause problems down the line. To really try to start applying paste, we generated a set of constructs that package it into a couple of different delivery modes. Because of the relatively large size of paste, one of the angles that we looked at that performed particularly well was packaging components into adenoviruses. The attractive thing about an adenovirus is that it has a very large cargo capacity. And so in applications for paste where your template may be particularly large, for example, outside of an AAV's carrying capacity, you could use an adenovirus to deliver a much larger payload. However, we were also interested in generating both RNA and lenti and AAV constructs, which we had some success with editing both in hex cells and in more primary cell lines. I'd like to just transition to a different part now, which is to say that we've been developing this tool and we'd like to try to apply it. So I'll talk very briefly about how we've started working on applying the paste system to a disorder that is potentially particularly well suited for it, which is cystic fibrosis. So cystic fibrosis is a disease that affects quite a large number of people, but not necessarily in the same way, in the sense that there are a very large range of genetic modifications or genetic mutations that can cause the cystic fibrosis phenotype. Generally speaking, cystic fibrosis usually comes from a mutation in a particular membrane channel that transports chloride ions. But the distribution of mutations that cause it spans almost the entire CFTR gene. And so this is a case where it may be particularly attractive to replace a non-functional gene and effectively address a whole range of patients with the same therapeutic, rather than trying to focus on a base editing or similar strategy that may work for a subset of the patient population, but not everybody. So what we've started looking at is trying to replace portions of the CFTR gene. And when I say portions, we're effectively replacing all of the exons of cystic fibrosis transmembrane receptor with a single paste cargo. And the way that we're going to do this is by inserting our payload containing the entire length of the gene into the first intron of the CFTR gene. By inserting the exon or the cDNA for CFTR and immediately upstream inserting a splice acceptor, we can encourage the natural exon 1 of CFTR to splice with our inserted cDNA and then ignore the remaining downstream exons. And so to start working on this, some other members of the lab, Christopher Fell and Dario Tagliaferri, have started trying to generate constructs that work for pegRNA insertion and then ultimately paste insertion at intron 1 of CFTR. And they've been doing this by tiling different combinations of pegRNAs to insert the ATB, or the first part of the paste machinery. And what we've seen is that while the CFTR intron is incredibly large, there are regions that are more or less amenable to insertion. And actually, by tiling different windows within these regions, we're able to find like a variety of different positions capable of inserting a CFTR payload at rates greater than 20%. And actually, by taking these regions and then applying some of the modified or engineered versions of PASTE, including by modifying the linker or removing the linkers between the prime editor and the VXP1 integrase, we can generate PASTE insertion rates of up to 60%. So this is actually really encouraging that we may be able to replace in a majority of cells or in a majority of loci the entire CFTR gene. So to move forward with this, we've been working with some collaborators to generate both RNA delivery of PASTE, so delivering the prime editor and the BXP1 as RNA, and then the template for insertion as a AAV or DNA template, and then introducing these to a mouse model of cystic fibrosis. So these results are unfortunately not back quite yet, but we're kind of hopeful that we'll be able to demonstrate some in vivo editing with this application. To quickly show some of the interim data, we've been testing PASTE for delivery of the CFTR payload in a model of lung epithelial cells from humans, known as 16-HBE cells, and can generate a low but appreciable rate of insertion using an entirely RNA system where only the payload remains DNA. The reason the payload has to remain DNA is because that's what BXP1 can recognize. So just to briefly summarize, PASTE, as developed, allows for the insertion of very large cargos without the generation of a double-strand break, unlike certain other CRISPR tools that rely on Cas9 cleavage or homology-directed repair. And we've been optimizing PASTE as a tool for a variety of different applications, but we've started taking it a little bit further down the road for the application of treatment of cystic fibrosis, which we think is a particularly valuable or particularly relevant target for PASTE editing just due to the nature of the mutations that cause cystic fibrosis being spread throughout the CFTR gene. What we're working on right now is trying to both improve the system's function in terms of overall editing rates, and then improve really within the models, both mouse models and human tissue model, both the delivery of the machinery and the overall editing. So right now, PASTE, in its initial form, is capable of very large integrations, integration of a wide variety of different cargo sequences as long as they have the correct ADB sequence, insertion of very large payloads, including therapeutically relevant ones, applications that require very specific labeling, such as tagging of proteins in frame, or insertion of multiple payloads into multiple loci simultaneously. However, we're still working on some problems related to delivery of these constructs, especially in vivo. and we require a DNA template. One of the other minor issues is that paste insertion will leave flanking at B and at P sequences, although this is less relevant in applications like the CFTR insertion, because we can simply ignore these and have them be skipped by the such that you recombine the gene with the splice acceptor. Moving forward, we're particularly interested in trying to find novel integrases that may have higher activity and also increase the prime editing rate through strategies such as designing smaller or higher affinity attachment sites. We're working on screening factors that improve efficiency within the cell through CRISPR screening, as well as modifications to the paste system or the arrangement of its components, and really trying to develop some more concrete design rules for the guide RNAs so that we can predict more easily what an optimal insertion guide would be. So I would just want to quickly acknowledge the work of some of the other authors of the paste paper, including Matt Yarnall, Rohan Krajewski, Lucas Villiger, Eleonora Unidi, Justin Lim, and Nathan Zhu, and also the members of the current PACE team, Christopher Fell and Dario Tagliaferri, as well as my PIs, Jonathan and Omar, and the Brain and Cognitive Science department here at MIT that hosts us.